you can hear me. My name is Conrad. I'll be your host for this last session of today, last but certainly not least. So now we're going to go down to the single molecule level. Now, first speaker will be Markus Schewe. He's currently postdoc in Thomas Baukrowitz group at the University of Kiel, so experts for potassium channels. And he will now share his insights on the gating of the TREK-2 K2P channel.
Yeah, thank you very much, Conrad, for the introduction and all of you that come in. And we have such a nice weather outside and after the lunch break. And as Conrad mentioned, now we switch gears and come to the smallest devices of non-iron. But at first, I want to introduce our lab also a second ago. Yeah, now it works. So we are a small ion channel group that is located at the Kiel University at the Institute of Physiology. So in a way, we are the outsiders there because we are mainly interested in the regulation and structure, function, and molecular details of pharmacological regulation of ion channels. So we don't work with animals and tissues. So we work with cells or even with ion channels that we synthesize cell-free and reconstituted in lipid bilayers or measure it on a porter patch on a single cell level. And one expertise of our group and that I want to show before here is that we combine functional and computational electrophysiological techniques to gain insight into the gating and the pharmacological regulation of our channels of interest. And as I mentioned, therefore, we use these small but yet powerful devices from non-ion. So what are the ion channels of interest in our group? So mainly we are interested in hyperpolarizing channels, mainly potassium selective ones, and especially in this small and latest identified group of K2P channels, so tandem-pore domain potassium channels. It's important that you don't think that there's two-pore channels that are two-pore domain potassium channels. So that's a little difference here, but quite important. And these tandem-pore potassium channels are thought for years to be simple leak channels that stabilize the resting membrane potential of cells. And in some, we now know 15 members of these K2Ps that can be divided by their sequence homology, but also by their regulation properties or by their main gating properties. And so today I want to refer to a mechanosensitive K2P channel that's called TREK, and want to share with you some of our new insights into the regulation of these channels. And yeah, in big contrast to that what David showed yesterday for piezo, that is a channel with only one gating stimulus and that can have major advantages. We have the opposite here. So the TREK K2P channel is a multimodal regulated ion channel. So when in the morning in your lab it's 2 degrees hotter, then you have a different activity of the channel. It's not so dramatic, but in the end, these ion channel is regulated by all physiological stimuli that you can imagine, like changes in the pH, temperature, lipid composition of the membrane stretch, partner proteins, phosphorylation, and so on. And we investigated some of these regulation mechanisms for years now, and described these mechanisms of regulation. And on the other hand, we also demonstrated in a few works how we can interfere with these evolutionary conserved gating mechanisms with small molecule regulators. and describe certain binding sites in these small proteins. And before I want to show you some new data, it's quite important to know what are the main gating principles for these ion channels. And at first, you have to know that these channels have only one major gate that resides within the selectivity filter. That's different to invert rectifier or voltage-gated K channels. So that was a work that was done together with Marcus Rapedius that time in Kiel more than 10 years ago now. And we figured out that there's one major gate in the selectivity filter that is also responsible for voltage gating and bears an unusual voltage gating mechanism. Further, these channels exhibit a very interesting gating hub, the C-terminus, a very long strip in the cytosol that functions as a sensory domain for pH sensing, temperature sensing, PIP binding, for example, and also for phosphorylation. And it's thought that attachment of the C-terminus to the inner leaflet of the membrane is associated with a more active channel, while the repulsion from the membrane leads to channel inactivation. And the third point, important to know in gating, arises from different crystal structures that were solved for TREK channels by our collaboration partner in Oxford, by Stephen Tucker's group and Liz Carpenter's group that time. And it was shown that TREK K2P channels can exist in at least two different states, an up state and in a down state. And these up and down configuration mainly arises from the movement of the transmembrane helix 4. And in the up state, this helix is more orientated to the membrane, and in a down state, it's more straight. And interestingly, this movement of this helix has a nice pharmacological side effect. When the channel is in the down state, the more straight transmembrane helix 4 opens side portals here, side fenestrations that can accommodate different compounds like the antidepressant norfluoxetine. When the channel is in the up state, the upward movement of the transmembrane helix 4 closes the side fenestration and the channel is insensitive to norfluoxetine. So we have a nice state-dependent modulation with these compounds here. And we validated these effect by different activatory mechanisms when we activate the channels by using rubidium as permeating ions. So that is a activatory mechanism that resides in the filter. So there are not big conformational changes. Then you can see that the norfluoxetine sensitivity stays the same. There's the same affinity in the activated rubidium state and in the basal potassium state. When we activate the channel with a molecule that binds behind the filters, described as an TREK channel activator, then these affinity for norfluoxetine drops. And that is in good agreement because the co-crystallization of MS335 with the TREK1 channel shows the upstate configuration of the TREK channel. And so we can use norfluoxetine binding to describe in which kind of state the channels are. So what we want to investigate now, or what is our main focus in the moment on, is how phosphorylation regulates K2P channels and what how the signal from the phosphorylation site in the C-terminus is transduced to the selectivity filter. And therefore, we at first checked if this gating principle with the movement of the C-terminus is correct. And therefore, we utilize the assay that we call the membrane tethering assay. We introduced cysteine residues in a regular manner in the proximal C-terminus of the channel. And then we attach to this cysteine residues different kind of chemical probes, MTS probes that bind irreversible to the cysteine residue. And once these probes have an acyl chain that's very lipophilic, or they have a hydrophilic head group like the positive or a negative charge. And what we found is we checked at first, of course, if when we apply these substances to the wild type channel, there's almost no reaction. But when we apply that to channels with this introduced cysteine residues, that in a very regular manner, we can activate these channels by this lipophilic decile MTS compounds. It seems that cysteines that are orientated to the inner leaflet of the membrane can be or results in activation of the channel by attracting the C-terminus to the inner leaflet of the membrane by these chemical probes. And that shows us that the channel must be in a state transition, or must be moved to the up state. And we verified it with the norfluoxetine assay again. So when the wild type shows normal norfluoxetine behavior or binding, when we activate the channel with the decile MTS, then we see that norfluoxetine sensitivity drops. That means attaching of the C-terminus to the membrane results in a down to up state transition of the channel. So how would that look like for phosphorylation? At first, that is what I mentioned. We also verified that with computational electrophysiology. So we attached to the TREK-1 channel the decile MTS or the MTSET probe again. And what we see within different simulation times here with 500 nanoseconds, that every time the channel with a decile MTS shows an attachment of the C-terminus to the membrane, while the charged probes move the C-terminus more away from the membrane. For the phosphorylation, we know two different phosphorylation sites in the C-terminus, the PKC site and the PKA site. And we know that phosphorylation or dephosphorylation would, in a way, attract the C-terminus also to the membrane or repulse it from the membrane. And therefore, we used phosphorylation mimics. That means that we mutated these two serenines to alanine or to asparagine. And what we see is when we mimic dephosphorylation, that means a state where the channel can attract the C-terminus to the membrane, the channel must be in an up state within these dephosphorylation mimics, because the norfluoxetine sensitivity was dramatically altered, while phosphorylation mimics that seems to attract the C-terminus should repulse C-terminus from the inner leaflet of membrane, even more increase the norfluoxetine sensitivity. This results also suggest that there's a more down state than from the crystallographic results so far. So at that moment, we were interested how these signals from the C-terminus are transduced to the selectivity filter. And that are the results from a PhD student that started her work 6 years ago, and nowadays we would like to upscale our NAN-ion devices to avoid such a I would say extensive work on different mutations. We checked 120 point mutations for the activity of the TREK-1 channel with these assays. So we activated the TREK channel by a two-step pH activation and quantified mutations that show a higher PO at pH 5 in comparison to pH 8. And when we show these or highlight these residues that show these altered activity of the channel or map it on the channel structure, we can see that they cluster from the C-terminus to the selectivity filter. And when we further investigate these high PO mutants with the norfluoxetine assay again, then we can show that all these mutations perturbate the downstate configuration of the channel and leads to an upstate transition that in a is correlated with a decreased norfluoxetine sensitivity. We confirmed that functional screen by a cluster analysis that we did in the down and in the up state of the channel, and we identified 84 short-range interaction of residues in the channel protein, and how this network works is written on another page. I want to focus on a very interesting point here, because this interaction network is interrupted by the small compound regulators that were described so far for K2P channel modulation. This is this ML335 compound that we is allocated here behind the selectivity filter, and the small molecule compound BL1249 that directly interferes with the transmembrane helix M4 filter interaction. And for this compound, several years ago, we co-crystallized this molecule with a TREK channel and show that it sits also in the side pockets, and that the tetrazoa ring of this molecule that is charged under physiological pH resides or points toward the iron permeation pathway of the channel, right to the selectivity filter. And by this orientation, BL1249 increases the potassium concentration under the filter, so it accumulates as this site, thereby changes the selectivity filter ion occupancy, what activates the channel. Interestingly, that was not a mechanism of activation that was limited to BL1249 that also worked for compounds that we heard yesterday or today. For example, this NS compound that regulates PK channel activation. HERG channel modulators utilize such kind of activation mechanism. And also these compounds have the same effect in the other channel proteins like hERG or BK. That seems to be a very evolutionary conserved mechanism. But we were still further interested how this molecule works, or how we can overcome such a polypharmacology of these small molecule regulators, and therefore we synthesized a BL molecule where we modified these negatively charged group to a triazole, for example, that's not charged at pH 7.4. And interestingly, we found that this compound has the same apparent affinity for TREK channels like the charged molecule, but has a reduced potency. And when we activate the channel with this BL compound, fully activate the channel, and then apply the one with the triazole ring, then we see that we can compensate or can reduce the tonic activation via BL1249, where we concluded that these compounds both compete for the same binding site. And further on, shows that also the interaction of the core body of the BL1249 with the transmembrane helix M2 and M4 should have a further component of activation onto the selectivity filter on TREK channels. And in line with that, we also try to find new modulators that overcome these BL1249 effect, and we screened a library of more than 500,000 compounds and identified 30 molecules that are proposed to be selective selective TREK channel activators, tested them against BL1249, and that was done with this new porter patch mini devices in our lab. And so we described these new TREK channel activators that further on always or still lack a selectivity filter, the selectivity for TREK1 channel. So there's still the burden of missing selectivity for TREK channel activators so far. And with this results, I want to close my talk and summarize the effects described today. So what we learned from our modification assay for TREK channel gating is that phosphorylation utilizes the C-terminus movement to the inner leaflet of the membrane, or the repulsion from the membrane, to result in a state transition from down to up state movement. And we identified the network that is responsible to transduce the signals from the C-terminus to the selectivity filter. And further on, we can describe how small molecules interfere with these network and TREK channels, and that these interference of these modulators use an allosteric and electrostatic mechanism, as well as an allosteric pathway that transduces via the transmembrane helix onto the primary gate in K2P channels. And yeah, with this, I want to thank our PhD students in the lab that mainly carried out the work here, and of course also all of the PIs that were involved in this long-term work, and of course all of you for your attention. Thank you very much.
Thank you so much, Markus, for this really great talk. Are there any questions? Not as far as I can see, so maybe to stay halfway on time. If you have questions later on, please approach Markus. So yeah, we could just go on with the next talk. Now it's